Assalamu alaikum, dear friends. In this video, I'm going to begin the heart physiology. In heart physiology, there is a topic and that is called intrinsic conduction system. I'll discuss the intrinsic conduction system, what actually intrinsic conduction system is, and what are the autorhythmic cells, how the electrical events takes place inside the heart, and what are their different areas of the autorhythmic cells found in the heart. These are to be discussed in this topic, so keep watching the video till the end, and then share these videos with your friends as well. So let's begin. The topic is here, I will mention, it's called the intrinsic conduction system. Intrinsic conduction system is the system of the heart. That means the heart controls its activity of its own. That means the heart shows the wave of depolarization of its own, or we can say the action potential is created by the certain kinds of the cells which are found in the heart by its own. That's called the intrinsic system of conduction. Or simply we say that there are internal factors in the form of autorhythmic cells which controls the activity of the heart by its own. That's called the intrinsic conduction system. Firstly, you must know that wave of depolarization is controlled by the heart of its own. Wave of depolarization, that is action potential of its own by the help of autorhythmic cells or non-contractile cells. Then I will discuss what actually autorhythmic cells are or non-contractile cells are. I'll discuss these cells later on. So wave of depolarization is created by the heart of its own. That constitutes intrinsic conduction system. Firstly, you must know that the heart work as a coordinated unit. It is only because of the presence of Gape junctions. Gape junctions are the junctions which are responsible to flow the ions responsible to flow the ions from cell to cell. Hence, a separate of information takes place from cell to cell. That means throughout the heart. The heart is also innervated with the help of nervous system, but it doesn't depend upon this. So neurofibers we say in the nervous system also alter the activity of the heart. Alter the activity of the heart. That means nervous system is actually the extrinsic factor. But here I'll only talk about the intrinsic factor in the form of autorhythmic cells or the gap junction. These autorhythmic cells, I will write here, AR cells have unstable resting membrane potential. Autorhythmic cells have always unstable resting membrane potential. It is said that this unstable resting membrane potential drifts towards the threshold. Constantly, we say that AR cells have unstable resting membrane. It constantly depolarize. It constantly depolarize and it drifts towards the threshold. Pacemaker potential, or it's also called the pre-potential. What is pacemaker potential or pre-potential? It is actually the membrane is always having unstable resting membrane potential. This spontaneous changing membrane potential is called the pre-potential. Spontaneously changing membrane potential is called pre-potential or pacemaker potential. That means, once again I will tell you, the AR cells, they are also called the autorhythmic cells. They initiate the wave of depolarization of its own. They have always unstable resting membrane potential. That means their membrane also gets depolarizes and it drifts constantly depolarizes and drifts towards the threshold. Actually, this having the pacemaker potential or the pre-potential, these are actually the spontaneously changing membrane potentials which drifts the membrane towards the threshold. I hope that you got this information. But the fact is there that it is not understood clearly, but it is believed that this unstable resting membrane potential is due to gradually reduced permeability to potassium ions. The membrane is having reduced permeability to the potassium ions, while as sodium ions entry takes place at slower rate. You must remember this point. The gradually reduced permeability to the potassium, but the entry of the sodium ions takes place, but at the slow rate. I'll make use of this chart here. So what happens? It upsets the balance between potassium loss and sodium entry is upset, and hence interior of the cell becomes more and more positive and less negative. The cell reaches to the threshold point. Ultimately, at the threshold, that is approximately minus 40 millivolt. That's approximately minus 40 millivolt. What happens at this point? So fast calcium channels opens up. Fast calcium channels opens, and there is an explosive entry of calcium. There is an explosive entry of calcium ions from extracellular space. The calcium channels gets open up, then the explosive entry of the calcium ions takes place from the extracellular, I have mentioned here E, extracellular space, and there is a little bit entry of sodium ions, minute quantity. Once again I will tell you what happens here. I'll make use of this diagram and the time taken in milliseconds. This is the point where reduced potassium permeability and sodium entry at slow rate. Sodium entry at slower rate. These 2 points depicts here. And with the help of this, what happens? Unbalance takes place, and when it reaches to the threshold, that's actually the minus 40 millivolt. Here is a threshold, that's minus 40 millivolt. What happens? Calcium channels opens, entry of calcium, that means calcium permeability increases, and here the fast calcium channels open. Once again, potassium permeability decreases, sodium entry at slow rate, it reaches to the threshold point. What happens at the threshold point? Fast calcium channels gets open up, there is an explosive entry of the calcium, and from the extracellular space, sodium ions also goes inside the cell, but at the slower rate. So it is actually the entry of calcium ions rather than the sodium ions. What happens? It rises the action potential. It rises the phase of action potential. What happens? Then there is a contraction shown by the heart. In other excitable cells, or we can say in other excitable cells, what happens to the other excitable cells? Now, there is a gradual this downward arrow indicates the decreased calcium permeability. The decreased calcium permeability and increased potassium permeability. there is a downfall of the graph and it reaches towards the or below the threshold point. And what happens? Once again I will tell you, calcium permeability decreased, potassium permeability increases. In increased potassium permeability, there is a wave of we can say the repolarization. When it reaches to the when the potassium permeability, increased potassium permeability reaches up to the repolarization, what happens? The potassium permeability once again declines. Then there is a closure of the potassium channels. When the potassium channels gets closed, what happens? There is a depolarization again takes place. It's also called the pacemaker potential. Again the pacemaker potential. Once again I'll tell you this graph. Here is a minus 60 millivolt. What happens at the minus 60 millivolt? There is a reduced potassium permeability to the membrane, and there is a little bit of sodium entry takes place, and it reaches. There is a, we can say, a slow depolarization takes place. It reaches to the minus 40 millivolt. That is a threshold point. And what happens then? There is an opening, or we can say the opening of fast calcium channels takes place. Explosive entry of the calcium ions from extracellular space into the interior of the cell. And then what happens? And the action potential takes place. That means the wave of depolarization increases. When there is the calcium permeability, when the calcium permeability decreases and the potassium permeability increases, there is a downfall in the graph, and it reaches once again below the threshold point. That means minus 60 millivolt. That means deep repolarization takes place. When repolarization takes place, then there is a slow depolarization once again takes place. When there is the inactivation of the potassium channels takes place, and it is also actually the pacemaker potential, because I have told you what actually pacemaker potential is. That is nothing but actually it is the spontaneously changing membrane potential. That's also called the pacemaker potential or pre-potential. I hope that you got this information. This was actually what actually the pacemakers do in the heart. I have drawn here the structure of the heart. There are autorhythmic cells. These are the cells which initiates its own depolarization, which are found in the heart. Autorhythmic cells, non-contractile cells, they are found in different areas in the heart. They are found here in the form of SA node, SA node. This is also called the pacemaker. It's also found in the form of AV node. It's called the pace setter. Pace setter. This is called the bundle of haze. And these are called the bundle branches. And these are called the Purkinje fibers. In the previous lectures, I had talked about the heart wholly and solely, so far as the anatomy is concerned. Here is the physiological events. I will use this diagram and then I will discuss all the points which I have mentioned here. Firstly, autorhythmic cells, they are also called the nodal cells. Autorhythmic cells in the form of SA node. I will mention here, this is the first point. SA node. It's also called the sinoatrial node. The mass of cardiac cells or the autorhythmic cells having mammoth function have a crescent shaped structure which is found in the right atrium below the entrance point of superior vena cava. Here is an entrance point of the superior vena cava where it is located as a patch of cells or the mass of cells. This SA node initiates the wave of depolarization, and help us to beat the heart at the rate of 75 beats per minute. 75 beats per minute when there is the presence of intrinsic factor in the form of autorhythmic cell. When the external factor comes into play in the form of neural and hormonal control, that is the extrinsic factor, and it may reach up to 100 beats per minute. It may reach up to 100 beats per minute. The SA node beats, and its beating is called the sinus rhythm. It actually, this also called the pacemaker. Why? It sets the pace, or it makes the pace of the heart as a node, and its beating is actually called the sinus rhythm. Note this point as well. Then here is another node that is called the AV node. AV node, also called the pace setter, it is actually called the arterioventricular node. It is found above the tricuspid valve, so it is also found in the right atrium. It's also the patch of muscle, and here you must know that the wave, or we can say the impulse, separates towards from the SA node. Mention here this point. With the wave of depolarization gets initiated in the SA node, it separates towards the we can say in both the arteria as well as towards the AV node, but it separates in both the arteria by means of Gape junctions. I have not mentioned here this point. We will mention here once again. SA node, from SA node, the wave of depolarization, spreads to both arteria via Gape junctions. Hence, arteria gets contracted. And the S in from this AV node, atrioventricular node, it gets information or it gets stimulated from the SC node where the depolarization wave has already created. So AV node is stimulated by SA node via internodal pathway. I mentioned here the SA node, internodal pathway as well as the AV node with the help of red marker. This is the internodal pathway, INP. Internodal pathway, through the internodal pathway, the message gets spread, or we can say the wave of depolarization spreads towards the AV node. What happens then? Actually, there is a 0.1 second delay at AV node. 0.1 second delay is there due to the presence of less Gape junctions. When the AV node gets stimulated, the information spreads towards the another this nodal fiber in the form of bundle of his. This is the third point I will mention here. Bundle of his, or it is also called the AV bundle. This bundle of his or AV bundle is found in the superior part of interventricular septum. This is the septum here, which bifurcates or separates the right ventricle from the left ventricle. This green line indicates the bundle of his, or it's also called the AV bundle, and it is short. The information separates from SA node to the AV node and from AV node to the bundle of his. When then bifurcates into two branches. So the fourth point is the bundle branches. Bundle branches are two, left and right. There are two bundle branches, one is right and another the left. These gets arised from the bundle of his and goes towards the apex of the heart through this septum, intraventricular septum, and then they turn to the what it called as the ventricular walls. So there are two bundle branches, one is the right and another the left, because these two branches goes towards the heart apex and then they turn superiorly and goes towards the upper parts of the ventricular walls. And the fifth and the last point, so far as the sequence of excitation is concerned, fifth point is the Purkinje fibers. Most important. Purkinje fibers. These Purkinje fibers are actually the fibers which gets arised from the bundle branches. These are the Purkinje fibers, and they get embedded, or we can say they penetrate into the ventricular walls. You must know that this is actually the pathway through which the atria and the ventricles gets contract, because there is no connection between the atria and the ventricles because they lack the Gape junctions. This is the only way through which the conduction, or we can say the spread of information, takes place from the atria towards the ventricles. And the sequence of events takes place like this: from SA node, then the AV node, then bundle of haze, then bundle branch, and ultimately the Purkinje fibers. Once again, I will tell you, these Purkinje fibers are more found in left ventricles than right one, right ventricle, because left ventricle is thicker than right ventricle. And these Purkinje fibers penetrate deep into the inside the ventricular walls, so that the contraction takes place. Again, in the ventricles there are the cells. What are they? Papillary muscles, papillary muscle cells. These papillary muscle cells are innervated with the help of the Purkinje fibers. They have more Gape junctions. Through the Gape junctions there is a full of ions, and the information goes toward the papillary muscles, and the papillary muscles in turn tightens the chordae tendineae. These blue lines here, string like lines, indicate the chordae tendineae. They get tightened so that they doesn't get what it called as the balloon back inside the atria when the ventricular contraction takes place. So Purkinje fibers are in one way or the other way innervates the ventricular walls. Here I have mentioned here are more Purkinje fibers in the left ventricle than the right one, because it has more thickness. So Purkinje fibers actually innervate the papillary muscles, and then the depolarization wave quickly goes from SA node towards the papillary muscles by means of Purkinje fibers. As far as the depolarization wave is concerned, the time of depolarization wave takes place from SA node to the last cell of the Purkinje fiber takes about 0.22 seconds or 220 milliseconds to reach here. You must note this point. From SA node to AV node, from AV node to the bundle of haze, from haze bundle pipe, these bundle branches, and up ultimately up to the last cell of the ventricular muscle fiber, what happens? It takes about 0.22 seconds or 220 milliseconds to raise the action potential, or we can see the ventricular this contraction takes place. Ventricular contraction takes place as it squeezes out the blood from the heart into the right ventricle, squeezes the blood out from the heart by means of ASLV, pulmonary similar valve to the lungs for purification, and this squeezes out, this is the left ventricle, is squeezes out the blood from the left ventricle into the main aorta to all cells of the body through this called the ASLV, aortic semilunar valve. So you must remember these points. Another point here, this SA node I have mentioned earlier, 75 beats per minute. Then the AV node decide to beat about 50 times per minute, and bundle, then bundle of phase, bundle branch and the Purkinje fibers is said to be beat about 30 times per minute. So once again, these are actually autorhythmic cells, show the sequence of excitation. If there is the absence of these autorhythmic cells, or the sequence of excitation doesn't take place, or if they are found but there is deformity in their action, what happens? The heart, the pumping action of the heart affects very badly. So you must remember that these all what it called the nodal cells should work appropriately so that they pump the blood throughout the heart, or they pump the blood into the heart and out of the heart when this what it called the pumping action of the heart takes place. I hope that you got the information regarding the these nodal cells, as well as the information regarding the membrane potential versus time taken. That means how the pacemaker sets the pace, and how the slow depolarization takes place, and how the action potential takes place. And also you must get the information regarding the how this potassium permeability gets decreased, and it reaches towards minus 60 millivolt. This membrane potential reaches towards the 60 millivolt. So it is all about in today's lecture. Hope that you got the information. Allah Hafiz.